Hello dear students, good afternoon. I am Dr. Mohammad Ismail, Department of Zoology, J.A.T Arts, Science and Commerce College, Malegaon. This is the second video on MCQs on biological chemistry for TYBSc Zoology students. And in this video, we are going to see some important MCQs on carbohydrate chapter. So let's start. Question number 1, dash dash of the following best describe pH, and the options are: option A, the pH of a solution is defined as the logarithm of the reciprocal of the hydrogen ion concentration, means H plus ion concentration of the solution. Option B, the pH of a solution is a measure of capacity of its content. C, both A and B, and D, none of the above. See, what is the question? Dash dash of the following best describe the pH. So the correct answer of this question is option A. The pH of a solution is defined as the logarithm of the reciprocal of the hydrogen ion concentration. The pH of a solution, aqueous solution, is the measure of how acidic or basic it is. The pH of an aqueous solution can be determined and calculated by using the concentration of hydronium ion in the solution. So the correct answer of this question is option A. Question number 2. The pH of pure water is neutral because dash dash dash. Options are: the pH of pure water is 7. Option B, in pure water, the concentration of H plus ion and OH minus ions are equal. Option C, water do not contain free hydrogen ion and hydroxyl ions. And D, none of the above. So the correct answer of this question is option B. Look, the pH of water, why is neutral one? So the main reason behind this is in pure water the concentration of H plus ion and the hydroxyl ions are equal one, same one. That's why the pure water pH is neutral one or 7:1. So the correct answer of this question is B, not A. The pH of pure water is 7. Actually in this question reason is asked why the pH of pure water is neutral one. So option B is correct one. Next one, question number 3. Alpha D glucose and beta D glucose differ from each other by position of hydrogen and hydroxyl on carbon numbers. Options are: option A 1, option B 2, option C 4, and option D 5. So which one among these options is the correct answer for alpha D glucose and beta D glucose differ from each other by position of hydrogen and hydroxyl ions on carbon number? So the correct answer of this question is option A. Look, alpha D glucose and beta D glucose is an example of anomers, and an anomer of a means carbohydrate only differs in its structure at the anomeric carbons. Anomeric carbon being the functional group of carbohydrate, which is usually the carboxyl group attached to it. Means in cyclic form, carbon number 1 is anomeric carbon atom, and the position of hydrogen and hydroxyl ion on carbon number 1 specify the alpha as well as beta D glucose. So the correct answer of this question is option A. Next one, question number 4. The two functional groups present in a carbohydrates are: options are: option A hydroxyl and COOH carboxylic acid, option B CHO aldehyde group and carboxylic group, option C hydroxyl group and carbonyl group, and option D aldehyde group and hydroxyl group. So the correct answer of this question is C, hydroxyl group and carbonyl group. Sugars or carbohydrates are two major functional groups. One is an either aldehyde or ketone. Both are collectively called as carbonyls. And another one is, second one is alcoholic group or alcohol functional group. So in the first option, means option A, two functional groups are given here, carboxylic group as well as hydroxyl group. Among these, hydroxyl group is correct one, but carboxylic group is not present. While in the second option, aldehyde group is also given here and carboxylic acid functional group is also there. So aldehyde group is at some extent correct one, but second option is also not fitted. Option C, look at the option C. The first one is hydroxyl group, which is correct one, alcoholic group, and the second option is carbonyl group. So option C is the correct answer. So the two functional groups present in carbohydrates are alcoholic group and carbonyl group. So option C is the correct answer. Question number 5. Hydrolysis of sucrose gives dash dash dash. Options are 2 molecules of glucose. Option B, glucose plus fructose. Option C, glucose plus galactose. And option D, fructose plus galactose. So the correct answer of this question is option B, glucose plus fructose. Look, sucrose is a table sugar, while lactose is a milk sugar. Lactose and sucrose are both common kinds of sugar that features prominently in a typical diet. Though chemically similar in many regards, our body digests them differently using different digestive enzymes. They also have different sources in food. So sucrose is made up of two monomer units. One is glucose and another one is fructose. So the correct answer of this question is option B. Question number 6. Dash dash on hydrolysis gives one molecule of glucose and another galactose. Options are maltose, sucrose, lactose, raffinose. Dash dash on hydrolysis gives one molecule of glucose and one molecule of galactose. So the correct answer of this question is option C. Lactose on hydrolysis gives one molecule of glucose and one molecule of galactose. Lactose is a milk sugar. It is also a disaccharide made up of one glucose molecule and another one is galactose. Next one, question number 7. The linkage present in polysaccharide is called dash dash dash. Means the bonds present in the polysaccharide is dash dash dash. And the options are option A, peptide linkage, option B, glycoside or glycosidic linkage, option C, nucleoside linkage, and option D, ether linkage. So the correct answer of this question is option B, glycosidic linkage. Look, there are different polymers and among these polymers, one is polysaccharide, another one is protein. So in protein, the monomer unit of protein is amino acids, and all amino acids are joined together by peptide linkage or peptide bond. That's why they are also called as polypeptide. So option A is the incorrect answer for this particular question, because peptide linkage is present in protein molecule, while option B is correct one, which is the main responsible linkage or bond between monomer units of polysaccharides, and option C, nucleosidic linkage, which is present in DNA, which is another very important polymer of our cell. And the last one is D, ether linkage. Ethers are a class of organic compounds that contain an ether group, an oxygen atom connected to two alkyl or aryl groups. They have the general formula ROR dash, where R and R dash represent the alkyl or aryl groups. So option B is the correct answer. Next one, question number 8. Which is correct statement about starch? Options are: option A, it is a structural polysaccharide. Option B, it is a storage polysaccharide. Option C, it contains beta-1,4 glycosidic linkage. And option D, it is a branched chain polysaccharide. So which among these options or statement is correct for starch? The correct answer of this question is option B, it is a storage polysaccharide. While option 1 is not correct one and option C and D is not correct one. So the option A, it is a structural polysaccharide. Starch is not an structural polysaccharide. Structural polysaccharides are cellulose, which is present in plant cell wall, while starch is a plant storage polysaccharide. And option C says it contains beta-1,4 glycosidic bond. It is also not fitted in this and not correct statement for starch, because starch made by glucose. The monomer units of starch is alpha D glucose, and the linkage between all monomer units of starch is alpha-1,4 glycosidic linkage. But cellulose is made by beta D glucose, and the bond or the linkage between monomer units of cellulose is beta-1,4 glycosidic bond. So option C is also not the correct answer one. And D, it is a branched chain polysaccharide. Look, starch is also made by two main components. One is amylose and another one is amylopectin. Amylose is a polysaccharide made up of several D-glucose units. They are linked by alpha-1,4 glycosidic bond. And amylose is soluble in water and can be hydrolyzed into glucose unit by the enzymes present in our saliva, alpha amylase. While the another important component of starch is amylopectin, it is also a polymer made up of several D-glucose molecules, and it constitutes 80% of total starch. And amylopectin molecules are linked by same one alpha-1,4 glycosidic bond as well as alpha-1,6 glycosidic bond. Means there is a small branching is present. It is also it is water insoluble component of starch. So option D is also not fitted or not correct for starch one. So the correct answer for this question is option B, storage polysaccharide. Next one, question number 9. Cellulose contains dash dash dash, and options are amylose and amylopectin. Option B, glucose units joined by alpha-1,4 linkage. Option C, glucose units joined by a beta-1,4 linkage. And option D, both A and B. So the correct answer of this question is C, cellulose contain glucose units joined by beta-1,4 linkage. Next one and the last question, question number 10. The change in optical rotation with time of freshly prepared solution of sugar is called. Options are specific rotation, option B, inversion, option C, rotary motion, and option D, mutarotation. I am repeating the question. The change in optical rotation with time of freshly prepared solution of sugar is called as, the correct answer of this question is option D, mutarotation. Look, mutarotation is the change in the optical rotation because of the change in the equilibrium between two ionomers when the corresponding stereo centers interconvert. Cyclic sugar show mutarotation as alpha and beta anomeric forms interconverts. That's why freshly prepared solution after some time gives some different optical rotation reading. So this phenomena is called as mutarotation. So thank you for watching this video. I hope it will help you in your examination. Thank you and best luck.